to this afternoon session at Track 7 on civil society and advocacy. The topic for our session is developing health solutions aligned to Africa's genetic diversity. My name is Soli Makohliso. I'm based at the Swiss Federal Institute of Technology, where I serve as the Deputy Director of the Central Tech Center there. I have a background as an entrepreneur for almost 20 years and was trained as a biomedical engineer. My interests are very much in the area of technology innovations and how we can leapfrog low and middle income countries to actually capture some of the emerging opportunities that are being presented by the fourth industrial revolutions and other technologies that we see these days. This afternoon in this session, we have three exciting speakers, and I will just make a brief introduction of their profiles. I will make it, as I say, I emphasize the word brief because each of them could actually cover almost 2 pages in terms of the achievements that they have. And our first speaker will be Ms. Delali Atipo, who serves as the Chief Operating Officer and Executive Director of an exciting company called 54Gene, which is a health technology company that attempts to address the need to close the gap in global health disparities by ensuring the inclusion of currently underrepresented African data in translational research, as well as clinical development and application of things such as pharmaceutical drugs, which we hope could lead to medical breakthrough and new healthcare solutions that are particularly geared towards the African population. And our second speaker is Professor Michelle Ramsay, who is the director of the Sydney Brenner Institute for Molecular Bioscience at the University of Witwatersrand in Johannesburg. She is professor of human genetics and the South African Research Chair in Genomics and Bioinformatics of African population. Professor Ramsay is past president of the African Society of Human Genetics and currently the president of the International Federation of Human Genetics Societies. And her recent awards include being the winner of the Department of Science and Innovation Distinguished Women Researchers Award. And our third speaker is Professor Raj Ramisar, who is a professor and head of the Division of Human Genetics at the University of Cape Town. He also serves as the director of the Medical Research Council, Human Genetics Research Unit, and Cancer's Colorectal Cancer Research Consortium. He's also the principal investigator on the retinal degenerative disorders major research project. Apart from being involved with several established research projects aimed at elucidating the genetic basis of diseases in South Africa, he's also very interested in setting up research into understanding the genetic basis of the more complex, common chronic disorders such as hypertension in our population. So I invite you all to sit back and listen to the brief presentation. Each of the speakers will make a brief presentation of about 10 minutes. And after that, we will open the panel discussion to the rest of the audience. And you may submit your questions via the chat session, and I will try and then pick up the questions from there and then communicate them to the rest of the speakers. Now, without further ado, I now hand over to Delali. The floor is yours.
Thank you so much, Soli. So good afternoon, everyone. Good morning, depending on where you are. As mentioned, my name is Delali Atipo, and I'm the Chief Operating Officer of 54Gene. For those not familiar with the company, we are a health technology company that's focused on building diverse data sets so that we can really look at how we power scientific discovery, as well as advanced diagnostic and better treatment outcomes within Africa, but even really just within the global population. And I'll share a little bit more around that a little bit later on. But getting to the heart of the discussion today around health solutions and really why we're really focused on genetic diversity as a whole. I wanted to share this slide as an opportunity to really talk about diversity in genomic data, and Africa in particular being home to one of the most genetically diverse populations, especially when trying to understand disease causality and looking at discovery for new medicines. One of the things that you find, and looking at most of the data that's used for research right now, as a few of you already well know, a lot of it comes from European ancestry, where we're really not really powering drug discovery, looking at much more diverse data sets like from within Africa, which happens to host a lot of rare variants that can be used to power drug discovery. What you see in the representation in some of these graphs here is that we see rare variation occurring on the continent because of the fact that modern man, essentially, as we know, evolved from Africa. And because of that, it hosts a lot of that genetic diversity. When we don't include populations from Africa, we're looking at empowering our research to do drug discovery, then we're looking at potentially suboptimal treatments that are developed down the road, efficacy and safety in Africans. The profile of what that looks like can be diminished. And so we want to make sure that we're, as 54 Gene, including a lot more of the diverse data sets that can really be used to make sure that optimal treatments down the road are made for the African population. And these are, again, just a few of the studies that have been out there, like the 1000 Genomes Project, and some sequencing efforts that have become a little bit more of recent nature. For instance, for those familiar with H3 Africa, they recently came out with a study last October where just looking at 421 people within the continent, there was much more rich genetic diversity, almost 3 million variants that had not yet been discovered in just that small subset, versus a lot of other European-centric studies that had been done where not as much variation had been found. Now, why is this important, right? Even with the few studies that have been carried out in African participants, we've already seen that there is much fruit to bear as it relates to having therapeutics that are developed based upon findings within variations in the African genome. One of the true success stories that are often talked about is within the PCSK9 gene and also the sclerostin inhibitors. So for those not familiar with the PCSK9 story, thinking about cholesterol levels, it was found that a variant within a woman of African ancestry actually was able to help with better control around levels of cholesterol. And so that actually led to a huge class of drugs in the cardiovascular space. And for instance, with the launch of Repatha, which is almost $1 billion drug today, if not already at that mark. And so just looking at that example, if we're able to really harness and look at where are there either interesting variations, new targets that could be discovered due to variations in African genetics, we could be really looking at new therapies down the line that would not only just impact Africans, but on a global scale as well too. In the case of MNT, we see that with osteoporosis, and this was actually a variant that was discovered within a particular population within South Africa, we were able to see a new launch of a drug just a couple of years back that also was quite powerful in treatment of osteoporosis. And so these are just two examples that when you have better inclusion and better diversity in data, then we're actually able to power a lot more drug discovery in the process, and therefore better treatment outcomes for our populations. Now, in terms of unlocking the potential, I hinted at this a little bit earlier on. Being able to look at diverse data sets is not necessarily something that was a foregone conclusion, as we know from before. A lot of our genome-wide associated studies, or GWAS studies, only really included about 2.4% of diversity from Africa. So again, as I mentioned, the majority was coming from the European or ancestry. So in order to help power drug discovery, there's actually probably a lot that's been untapped that we're hoping as 54 Gene and a lot of other either academia or industry efforts are around this to have better inclusion of African data to help power through better healthcare solutions. Now, the way that we're particularly doing this as a company, just as an example for how we're trying to look at developing healthcare solutions, is around our capabilities and our infrastructure, and the fact that we're actually doing this on the continent. So what do I mean by this? In terms of being able to, first of all, generate the diverse data sets that we want to be able to see if we see any new novel insights, any sort of interesting targets that could be in certain disease area and categories, we decided to actually start building and working in partnership to develop disease cohorts. And that started principally with our biobank. So we are, one of where our regional headquarters is in Lagos, Nigeria. And so we've built out a biobank that has the capacity to be able to store biospecimens of up to 300,000 samples. And we work with different research communities, as well as tertiary institutions and hospitals to be able to collect around different disease areas. So primarily around non-communicable diseases. And the reason why we actually focused in on non-communicable diseases first is because we did feel that that was quite a neglected area. When often we talk about what are the huge disease burdens within Africa, we typically focus on TB, HIV, and malaria. But NCDs in combination have a greater disease burden than those three combined. And so within our biobanking process, we've been able to collect around neurodegenerative diseases, cancer, sickle cell disease, cardiovascular, et cetera. And we've been collecting that as part of our biobank and having that infrastructure powered using solar to help with the power infrastructure challenges that we know exist, not just within Nigeria, but other parts, to help make sure that our biobank is stable, but then also collecting that with the phenotypic information. We've also in our own labs, not only just collection of the data, but also from a technology perspective are doing the sequencing on the continent as well too. So really trying to get away from sending samples outside of the continent. And as 54 Gene, we're also training molecular scientists to be able to do whole genome sequencing, to be able to do genotyping, having the latest Illumina technology available at our fingertips to be able to have this capacity within the continent. Also, in addition to, besides just a molecular genetics component, is that we have a diagnostics infrastructure in place. And so as 54 Gene, we're focused in on 3 areas. And I've kind of touched within biobanking and our genomics capabilities that really helps to power our drug discovery. But the second part of what we also do is clinical diagnostics as well too. And so we have a diagnostics infrastructure that's looking at how do we have advanced testing. So doing our PCR-based technology, doing our histopathology, digital pathology, and having that infrastructure available at our fingertips, not only to power research, but also to power clinical diagnostics. And then on the clinical front, as I mentioned, we work with a number of institutions across the board. And from a data science perspective, once we've been able to generate the data, how do we now also have the in-house capability to do the scientific analysis, power the GWAS, power the PheWAS, so phenotypic wide-associated studies to translate eventually and be able to see if we can identify targets, validate those targets, and see which targets might be of interest to potentially carry through to the clinic. And this is just a quick sampling of, as I mentioned, of our biobank that I talked about a little bit earlier. We look at clinical and phenotypic data that's captured on disease characteristics from family history, comorbidities, history, different types of socio-demographic information, all looking at different sorts of social determinants of health. And I mentioned a little bit around some of the disease areas that we capture as well too. Ultimately, at the end of the day, as 54 Gene, and in terms of trying to drive health solutions, we really do believe in building the ecosystem within the continent. And I mentioned already around the infrastructure and investment that we've put. In addition, because we know that capacity in terms of the molecular scientists that we're using to do the work, from the academic researchers that we're working with, we're also trying to make sure we foster job creation. We're also trying to make sure we use sustainable solutions, as I mentioned, around solar. And then we're also looking at ways for which there's benefit sharing around the ecosystem as well, too, and doing that in an ethically responsible manner, from how we do informed consent to all the way to how we get ethics approval at the national level, so both the patient as well as through the institutional to the national level. And so this is just a little bit of a glimpse of how as 54 Gene, we're trying to drive healthcare solutions based upon driving better genetically based drugs and therapeutics for better health outcomes for all. And with that, I'll turn it back over to Soli and look forward to questions and comments later on.
Thank you very much, Delali. Thank you very much for such an exciting presentation. And so now we'll hand over the floor to Michelle. Over to you, Michelle.
Hello to everybody, and thank you so much for inviting me to be part of this session. Delali, that was really fascinating and incredibly interesting work that 54 Gene is doing. I've decided to focus my short 10 minutes more on Africa's genetic diversity and maybe just giving the participants some kind of sense for why we have this tremendous diversity and how important it is. So when we think about Africa as a continent, it is hugely diverse, and there are 4 major language groups. But what has really shaped diversity on the continent is the migration of populations. So we can look at this in terms of out of Africa migrations, 45 to 70,000 years ago, but then also looking at migration across the continent, and this having led to populations admixing and moving about. And this, of course, shapes the genetic diversity as well. And then the last few hundred years, migration out through the slave trade, but also back migration from Europe. So when we think about genetic variation and its impact on health, I think what is really important to understand is that when we think about health solutions, it's not only about the genetic diversity, it's about so much more. So for example, we have so many environmental influences on our health, and we have host factors which include our choices in terms of lifestyle that of course affect our microbiome. But we also have changes that occur in the epigenome, which are changes that don't change the DNA sequence, but changes how genes behaved and are expressed. And that gives us differential gene expression. And then layered into that complex mix is also the genomes. So all these factors together are mediators of the phenotype and put us on that trajectory as individuals from health to disease. So I think that's important to understand. So this is the paper that Delali also referred to, which was published by H3 Africa, as you can see here. And from sequencing a mere 426 whole African genomes, 3.4 million novel genetic variants were identified, most of them rare, but they give us an incredible opportunity to understand more about the biology or the impact of genetic variation on disease and coming up with health-related solutions. So when we look at Africa, as I said again, it's a very diverse continent. It always is to me interesting that people just talk about, solutions for Africa. Well, I think we need to think of that as many different solutions for different ethno-linguistic groups, different countries in the continent. And these are the different places in Africa that was sampled for the study. And you can see a predominance of samples coming from West Africa, not very much from Central, South, and East. So there's very much more that we need to still unearth and mine. When we look at the principal component analysis on the bottom here, based on the genetic variation, the point that I really want to make is that although there are discrete clusters that are different ethno-linguistic groups, what we could also see is that there's almost a continuum, so that neighboring countries have admixture. So genetic variation doesn't go along country lines or political borders, but really more in terms of populations and their associations with their neighbors. So this is a review that was published in May this year, really talking about African genetic diversity and adaptation and how this has actually informed a precision medicine agenda. And this brings us again to the fact that when you look at Africans, relative to our reference DNA sequence, they have so much more variation. So if you look at a group of European and Asian populations and individuals, they on average have about 4 million differences from the reference genome. But when we look at Africans, they on average have 5 million differences from the reference genome. So an order of magnitude more in terms of genetic variation. And when we think about phylogenetic analysis, that variation also plays itself out on the African continent and showing just how little of the variation is actually contributed by non-African populations, and bringing us again to the fact that most of the research has been done in non-African populations, and we have to be very careful about how we extrapolate that to African populations. And what you can see again here is the tremendous diversity when you are looking at different regions within Africa, and you can see how deep some of the evolutionary roots are there. And then lastly, when it comes to using polygenic risk scores to assess the genetic risk for a particular trait, if that research is done based on a discovery population from Europe, and you then apply it to European, Asian, and African populations, what you can see for a number of traits here is that they are much more predictive of what is happening in Europe than they are of what is happening in Africa, shown in the orange here. So there are many challenges that we have in Africa, I think, in terms of trying to translate this tremendous genetic diversity into practically useful, clinically applicable tests. So some of these challenges include, of course, the increased burden of disease on the continent, both infectious and non-communicable disease, and the fact that we have this tremendous heterogeneity, which doesn't only talk about genetics, but also about the environment. And then generally our insufficient infrastructure, the fact that we need more skilled people in Africa to do this work, and the fact that we just don't have enough data. But, we need to think and focus about what are the enablers of precision medicine approaches in Africa. And I've put right at the top here the issue of political will, that you need to want to do it and for people to invest in it for it to happen. And I guess this is part of what this meeting is about, is trying to build this political will. But of course, we need good governance and resources. We need a lot of data, and we need to think about sharing that data, both within the continent and globally, for that data to be used so that we can also develop tests eventually that are applicable to African populations as well. And then you can see all the other important things that we need. And right at the bottom here, digital technology, because that certainly is one of the drivers of innovation, is being able to access data. In the last column here, it's really about thinking about what the benefits are of doing genomic research in Africa, and that the benefits accrue both to global communities and to African communities. So when we think about the benefits of African-based research or research on African participants for the global community, it's about adding value and diversity to databases. And sometimes it is also that we can provide from Africa treatment-naive participant groups, when those are contrasted in much of the Western world where people take so many different medications. Then in addition, it can lead to the identification of causal variants, as well as novel biological discovery, and of course will be applicable to the large African diaspora. When we think of benefit to African communities, that would be directly in terms of diagnostic and treatment capacity tailored specifically for African populations. and then about knowledge of genetic predisposition to disease, taking us to those polygenic risk scores, and eventually really understanding the key gene-environment interactions that are so important in terms of disease. So this is a sort of a schematic looking at what is happening on the continent at the moment. And this is a review that's also just been published electronically and will come out in print soon. But it's really about trying to understand this translational bridge between genomic research and then taking us to clinical genetic services, and understanding that, you know, initiatives like H3 Africa is really enhancing what we see here in terms of genomic research in Africa. But that when we think of actual application and implementation, there's much less of that on the continent. And part of the challenge is that this has to happen in the context of a broader healthcare system, where you have like a huge clinical network, because of course, genetic diseases transcend, you know, most of the medical disciplines, and it's necessary to have that backup. So we in this article make an argument that there are 4 areas that can act as accelerators for translational genomics in Africa. And that is about capacity and infrastructure development, but also about having dedicated funding and support for this kind of work. And then, you know, coming back to this issue of political will, but also having supporting legislation and policy development that will underpin implementation. And then finally, the importance of communication, public education, and awareness. So this is the last slide, just talking again about challenges and opportunities. So I hope that, you know, we're beginning to make it clear that research really has to be done in Africa, on African participants on the African continent, to make sure that it's going to be relevant to Africa. And we have to ask the question about who will support this. Is it sustainable for it to be supported by American, European funding agencies, or do we need to look within the continent? Then we need innovative solutions, and these are critical, and they need to be tailored for the African continents, and furthermore have to be affordable and scalable to really make a difference. So again, making the point that we need different solutions for different countries depending on their context. And this was a
recording in progress.
Has a very good reason to be in existence. So all of this is just tying in very well. And now we have last but not least, Raj, and the floor is yours.
Thanks very much, colleagues. It's a great pleasure to be addressing this audience today. So thank you very much for the invitation, colleagues. So I head the Division of Human Genetics, which is involved with genetic service delivery, has a very strong diagnostic laboratory basis, as well as a research environment. So in terms of this particular audience, I think the advantage we can speak of with regards to genomics is that, you know, when we think about the 4th industrial revolution and the healthcare, we can literally think about us looking to the heavens, where we do a lot of astronomical research, and there's a huge amount of computational infrastructure that has been invested in. And when we're thinking about dealing with human biology and the human genome, you can think about us looking, in fact, in the other direction, deep within ourselves. And that's sort of a very nice philosophical picture with regards to looking outwards and deeply within ourselves, but where the technologies are exactly the same, i.e. dealing with vast amounts of data. So I just want to go to, and in fact, I loaded this slide just as our colleague Delali was talking about sclerostin inhibitors. So in fact, the disorder was discovered here by my predecessor at the University of Cape Town, Professor Peter Byton, when he described this disorder, Sternostiosis. And I'm not going to go into describing the disorder. And in fact, we had done a substantial amount of research towards identifying the gene. But towards the end, for some silly reason or the other, we handed over pretty much all of the IP to a company from Canada, and it completely slipped out of our hands. And if this turns out to be a blockbuster drug, then you can imagine we wish we would have learned a lot more about protecting IP at that stage. So that's just...
In this example, I will speak to you about Sternostiosis, and I'm asking you to please read up briefly about this condition. It was first identified in South Africa by...
Sorry, colleagues, I'm getting feedback. So if I can move on, my own work has got a lot to do with familial cancers, and we've done a huge amount of research where we've identified many different genes underlying familial colorectal cancers specifically. And we're now able to predict in these large families and often very extensive genetic lineages, those individuals who may be predisposed to disease, i.e. not yet affected, but will be affected in future. And this is illustrated in the family tree I've got here at the bottom. And this is quite remarkable. However, we've not made a concerted effort. It's being done right now with regards to literally taking this kind of diagnostic work nationally. And I'm really glad to be working with colleagues all around the country in developing a national familial cancer register. So one can actually understand that, while we're doing a lot of research on the complex disorders, like Michelle has been talking about, my own work on the genetics of psychiatric disorders, more recently genetics of schizophrenia in the closer population with our colleagues in psychiatry, is producing out some really nice work. That's quite far away from being taken into the service environment. However, what I wanted to emphasize in this talk is that we've got quite a lot of work around the departments of genetics around the country, including Johannesburg, Durban, the Free State, where we have enough genetic information on our populations to be able to introduce comprehensive genetic services nationally. One obviously needs political will in this regard, and as you've heard previous speakers talk about, and particularly in the development of policy alongside. So as I'm saying, we've got information on cancer, familial cancers, where we can actually intervene and prevent individuals suffering and dying of these diseases. The other area that we've expended a lot of effort in and that the facilitator introduced was in the area of the genetics of disabilities. And in this instance, apart from looking at the genetics of deafness and crippling disorders, we've developed a comprehensive portfolio of research on the genetics of blindness and specifically inherited retinal diseases. In terms of talking about genetic diversity, we know that these disorders are caused by mutations in more than 350 different genes. And we've done extensive work in our immigrant populations in the first instance, simply because we had material easily ready in that. But in the last 10 to 15 years, we've been looking at our indigenous populations, the Xhosa and the Zulu-speaking populations here in South Africa and others around the country. And in fact, what we can report is there are remarkably new findings in these particular disorders, i.e. we're discovering new genes and completely new mutations. The big deal about this is that we already have a big library.
Inherited from the blindness here.
We have comprehensive libraries of these individuals. And the slide here illustrates, that we recruit families from all around the country. The right-hand map shows families in whom we've actually identified mutations in specific genes. So there are large numbers of families in whom we've identified mutations and genes. And the big deal about this, is that this leads us immediately to the environment of gene-based therapeutics. So we have several hundred families already in whom we've identified mutations. And the big deal here is if you do not identify the mutation, and this has to do with the diversity of our populations, those individuals are not amenable to the remarkable new therapeutics that are coming out in this era of precision medicine. This little photograph at the bottom right illustrates simply the trains at one of the stations in Paris. What I wanted to show is that, in fact, if you don't have a ticket specifically with your specific disease and the specific mutation that causes the disease, you're likely to end up getting the wrong kind of therapy if you're going to access therapies at all. So the important thing is to know exactly what that individual's disease is, very important clinical or phenotyping information, more importantly, what is the genetic basis of this disease? Because with that information, it's literally like having a ticket to get onto the right platform and to get to the right destination to have the disease repaired or in fact prevented from developing at all. So this work is well on its way here in South Africa, and I'm very pleased to say that, you know, we could be carving out pathways, you know, where we map all of the research that's going on in the complex disorders, but at the same time be focusing on the disorders where we have this kind of really good information to be able to provide genetic services and therapeutics.
This slide,
so colleagues, I'm close to finish. So when we're looking at genetic disorders, on this slide, what I'm trying to illustrate is when we're attempting to identify what the genetic defect is, the implications are several. The first one is, as I indicated for the colorectal work, is to provide diagnostics and to have preventative interventions. On the right-hand side, you notice that there are therapeutic developments potentially that map onto the specific disease, the specific gene, and the specific disorders. Another colleague of mine, Colin Masembrembo, who's attached to Michelle's Institute, in fact, gave a talk this morning on pharmacogenomics. And the big deal here is, I'm not going to say anything about what he said, but simply to accentuate that, you know, it's all very well us providing drugs that are developed for disorders that are diagnosed in Western populations, because that's where the drugs are developed. But the reality is the genetic variation carried by our populations, firstly, in terms of what causes the disease, may be quite different. Secondly, in terms of whether that drug is actually processed adequately and appropriately, the pharmacodynamics and pharmacokinetics may be very different by the time the drug even gets to target what the actual problem is. So this is a hugely exciting area of work and there's a lot of work going on in this regard across the African continent, and especially through colleagues who are working on AIDS-free Africa. So colleagues, in my final slide, I'm simply addressing that the issue of partnerships is hugely important. So in this regard, I've got the government at the top. We can't always rely on government, but we know that we have quite a remarkable Department of Science and Innovation, rather than technology. But I'm not sure we adequately talk to one another as the DSI, Department of Higher Education, Basic Education, Economic Development. I really do think there's a lot more, and especially the Department of Health being brought into this cluster. Another facilitating entity we have is the National Health Laboratory Service, which is part of the Department of Health and has a remarkable national footprint in terms of understanding epidemiology of disease. And then we have the South African Medical Research Council and other funding agencies such as the National Research Foundation and so on. I do think we have got many things in place, but I'm not convinced that we're always talking to one another in terms of taking us appropriately towards you know, the next big frontier, which is the fourth industrial revolution. Colleagues, my final slide, this is a piece that I wrote a couple of years ago for Nature, and I called it African Dawn. I really do believe we're at a very important juncture where we've seen really from pretty much very little genomics research being done on the African continent to where there is quite a huge amount of genomic research, especially fostered through this entity called AIDS-free Africa. So rather than us being seen as the dark one, I really do think that the sun is rising. The biggest deal here is with limited resources, we've got to be clever enough to connect. And connecting the dots and being connected as human beings across different disciplines is really what's going to get us to make the difference. Chairperson, thank you very much. And I thank you for asking me to talk amongst these colleagues. Thank you.
Thank you, Raj. And that was, again, this is exciting in terms of how these three different talks actually, they actually speak to each other in some ways, because there we are trying to really see some concrete, you know, research projects that are illustrating, you know, with concrete examples on the ground. And now we, I think, what's nice as well here, and I'm picking up on your last topic, especially Raj, where you say we need to be speaking to one another, and I think one probably one major player that you probably forgot to mention there is also the private sector, because you were talking about the needs to be able to generate applications that can be therapies, diagnostics, etc. And that is obviously the primary forte, obviously, of the private sector once the research has been completed within the academic institutions. So, but very welcome, and indeed, under these circumstances, with the minimal resources we have, as they say, unity is strength. And on that note, let's try and see if we can open the panel for discussion. And I don't see any questions yet on the chat, so please don't hesitate to, anyone in the audience to fire on some questions here. But I maybe will try and, I've got a few questions of my own. And I think also what might be helpful, given this complementarity that we have in the panel, it's also quite, acceptable, and I would encourage it that even the panelists can probably ask questions from each other, because I think, you are all coming in, you fit in different parts of this very complex puzzle. So please also do, prepare any questions you think might be of interest that, from what you've heard here. And if I come back, just one of the things I was, when I was listening to 54 Gene and all their pillars, so to speak, one of which was, biobanks, and genomics, diagnostics, etc. So, and biobanks is a huge intellectual property, because that's where it all starts. Having access to those biobanks, is essentially intellectual property. You're studying intellectual property, and I'm curious as to. I heard from your presentation that you have established one, but it was only in Nigeria. Do you have any other, plans, or do you actually have any other facilities or biobanks being established in the different regions of Africa, and especially, hearing, the talk from Michelle in terms of this, significant biodiversity, so to speak.
Yes, thank you, Soli. And I apologize in advance if you hear my little one in the background who's just woken up. It's an exciting time in our household. So I'll attempt to talk over his screams. I apologize.
Yeah,
but one of the things that I think to keep in mind, and maybe Soli, if you could.
Okay, maybe we can come back to you.
I'll come back to the question.
Yeah, we'll come back to you. So let's yeah, I know this is the days of home office, so to speak. Now, I just wanted, maybe for the benefit of the audience, just to maybe speaking in very general terms, especially how I'll be curious to hear how you both reflect. I mean, back in the day, we started with, I think in the late 90s, we had the Human Genome Project. And there's lots of excitement. This is what opened, we started hearing the word high throughput. It was one of early days when we started hearing words such as high throughput as everyday tools in biology, so to speak, at least in research, being brought to the research domain. And those translated now, and then the genome was sequenced, etc. And then we had proteomics, metabolomics, etc. And I'm just wondering, how do you position, where we are and some of the work that we're doing now within that, within that continuum, so to speak. And another, I'm also, since we are trying to talk about precision medicine, and there was a time when we were talking about personalized medicine versus precision medicine, and I'll be curious if, since you are experts in this field, if you maybe can help clarify even to myself, I'm not sure, probably on a broader, there could be an interest in that.
So, Soli, shall I give that a first go?
Exactly.
So thank you for that question. And, I think the world is making strides in so many ways in terms of the multi-omics approaches. But again, we understand that most of this research is being done in the Western world on people of European origin. And on the African continent, we are now seeing a bit of a groundswell and more work being done, certainly in genomics. But I think that we don't have nearly enough data in the other omics areas. So when we look at databases and we try and interpret African variants, we're often still interpreting it in the context of what is known in the rest of the world, but not Africa. So I think data is so important, and we've got to generate it here on the continent. So I think until we do that, and we do that at scale, we're going to still be left behind. So I think somehow we need to galvanize an increase in effort and an increase in data. But not only that, we need processes and governance frameworks to actually share the data. Because if everybody generates the data, but they keep it for themselves, then you can't actually put it together, connect the dots, and really make something meaningful. And I think in terms of the translational process, we also have to understand that research is just the beginning, but there's so many steps that have to happen. But often it can be like the kernel that starts everything going. And Raj, just going back to what you were saying about the family, or with this very unusual bone disease, sort of leading to the discovery of a variant that could then eventually identify drug targets and develop a drug for osteoporosis for the rest of the world. And I think there must be many more examples like that in Africa. So we need to do the work here so that we can make sure that they make a difference.
So if I can add to that, Soli. So, obviously we wish, and I apologize, much like the baby crying, we've got building work carrying on at our institute, and there's just a huge amount of noise. So I apologize if the noise is coming through. But, there is a keenness to do pretty much everything. But the reality is the biggest deal is making sure we gather really good biological material. And that process has begun, as you've heard of the biobanks and so on. So there's a systematic process of gathering really good phenotype material. There's a concerted effort to just sample populations and get an idea of genetic diversity and variance across the African continent. So that's really good basic work. And the reality is that a lot of the omics work that is going on, whether it's metabolomics or exposureomics or whatever you call it, there's tons of that going on. A lot of that is still very noisy. There's a lot of data being gathered and a lot of filtering going on. There's some really nice work coming out of that also, but it's not something that we have to get in there. Some of our younger colleagues have left the continent. They're working on this kind of thing, and in due course they're going to come back. But it's going to be almost like cell phone technology. It moves so rapidly with informatics being what it is, and digitization and so on, moving in the right direction. I reckon that with us doing a lot of the basic foundation work, we are going to be in a really good place that when a lot of the stuff technologically starts to make sense, the noise is filtered out, etc. This is going to happen fairly quickly, like how quickly cell phones have grown, even on the African continent. We'll be right there with the material and well-gathered biological, clinical information, biological material, and so on. But absolutely, I think there needs to be a plan with regards to putting all of these things down. Yeah. Thank you, Soli.
Yeah. Thanks, Raj. Delali? Now that you've managed to...
In the era of COVID, you just have to be flexible, right?
He wouldn't want to miss out on this exciting...
Exactly. So I'll try to backtrack to the original question that I believe I heard. So just forgive me if I missed something. Feel free to re-ask it. But you were asking around, where does 54 gene currently have our operations? And yes, we are predominantly within Nigeria, but we're actually in the process of expanding our work at the moment. And so we are actually talking quite actively with a few partners in East Africa, as well as other places within Western Africa as well. And, I think to Michelle's, some of her slides earlier as well, too, I mean, it makes a lot of sense. We have to look at diversity across the continent. It's not just about one singular place. But one of the reasons why we did start in Nigeria was given the quite vast, diverse opportunity, you could say, within the country, given that it has around 500 plus or so ethnic linguistic languages. And so Nigeria is thought to have about a quarter of the ethnic diversity that we see across Africa. And so it was a natural place to start for a lot of reasons. But when you put that in context and look at the fact that when we were talking about northern Nigeria, southern Nigeria, or even other parts within certain same regions, the level of diversity that you find there is quite unique compared to some other places. But hands down, there's a lot more to be told when you look at other regions within Africa. And I think as well, yeah, go ahead. There's probably more I want to add to that later, but there was a question I think Michelle also had too.
In fact, there's a question in the chat which is sort of related to that, which says, how does your 54 gene work with the Nigerian, how does 54 gene work with the Nigerian public health sector? How would you do this in other African countries? So I guess, you're not there yet, but yeah. But
we're in the midst of it right now, so I can speak a little bit to it. But at least starting with Nigeria, and Michelle, great question. It's definitely been a model that's evolved over time. But as I mentioned a little bit earlier, even when we're doing our cohort building, we work with a lot of the tertiary institutions that are already in country around doing both institutional-based recruitment as well as community-based recruitment. And in our ability to do that, we also have our data sharing practices and want to have the ability to have co-publishing opportunities with our academic researchers as well too. So one of the ways we work together is partnering and providing our own operational infrastructure along with the operational infrastructure that the institutions have in place. But a lot of it, we obviously fund as 54 Gene, but we work within the parameters and the processes of the, as I mentioned a little bit earlier, the informed consent processes that we need to put in place, the IRBs, the national ethics, and ensuring that from a regulatory perspective, how are we making sure that we have, for instance, the right data access and biobank access committees in place. Some of our scientific steering committees that we put in place, we also make sure that we have representation from the community, as well as the researchers that we're working with. And so from an institutional reflection and working with the regulators, because also genomics to a certain degree, right? Though probably not for any of us on this call, it's still a relatively new space, right? Within the scientific community within Africa, right? So there's also an educational process, and we also try to do capacity building workshops in partner collaborations with our researchers to educate the regulators, making sure, for instance, from a data perspective, because as a company, we hold ourselves to a high degree and standard to not only just follow GDPR, but how do we also help to inform the Nigeria version, NDPR, with having the right data protection in place for participants that are participating in our studies. So there's a variety of different ways that we get engaged and involved, but in even engagements that we're doing within East Africa and looking at in other parts of West Africa, we're trying to ensure that we work with the local societies, as well as, again, the model of how do we work with the public institutions to be a part of our research processes and our operational processes as well.
Yeah. Thank you, Dilari. There's a few questions that actually, I guess, Michelle, asked another question from you. But before, maybe I'm curious, because before we come back to you, we will give you a break. There's probably maybe another question that's related to that, because Michelle, you advocated a lot for data sharing, and I'm curious, especially within this climate of suspicion by African governments of being robbed of data that emanates from the continent by the Western world. For example, in South Africa, I heard that there are intentions to try and limit the ability to export data, or at least in Tanzania as well. That data that is collected within the region may not leave the country. I think without, I don't know if there are permissions or etc. that are required. So in other words, there is the problem of trying to balance that prior exploitation that was referred to, for example, the loss of, there is a long history of unfortunate loss of intellectual property, to the rest of the world. How do we balance that? How can this be done at the same time safeguarding any intellectual property that emanates from such data?
I'll have a go at that, Soli. And it's a hugely important question and issue. And I think again we're seeing diversity across the continent because not all countries have data protection laws. And some have very stringent ones that may sort of stymie open science. So I think we really need to balance, the open science with protection of data. And you're quite right, in South Africa at the moment, we are grappling with research in the context of the Protection of Personal Information Act, and whether that may or may not limit sharing of data. But I firmly believe that if we don't share African data, Africa will never benefit, because it has to be compared to other data. It has to be used and mined and, looked at more carefully. So I think, the solution is not to keep it in one place, but rather to have a responsible governance framework in terms of sharing it, and then ensuring that there is some benefit that comes back to Africa. So I think the way that we share it, we have to frame, we have to have data access agreements, and people need to respect that. But it is a very tricky thing when it comes to intellectual property and so on. It's a hard balancing act.
Yeah, completely agree, Michelle. I don't know that there's anything else that I would add to what she said. I mean, as a private industry player, right? Obviously, there can be, the comments or thoughts around suspicion, as mentioned a little bit earlier, in terms of what's going to happen with the data, which is why we've been really, really mindful to make sure we have these data access committees in place within our own organization, and working and having actually external folks sit as part of our committees. Now, at the end of the day, we are still a commercial entity, right? And so we want to make sure that we have commercial rights to be able to further research along.
Sorry, I just wanted to add something. That it's obviously there's a tension between, we know that, you've got data crunches internationally, whether we're talking about Asia or Europe or the geographic north, where you literally, what we're going to do in a year, I mean, they're going to crunch through a weekend and get through stuff and either publish or develop property that we wouldn't even have seen. So there is a tension. But I think if we properly safeguard without just holding on to the data, but have conditions attached to discoveries coming out of this data, need to be handled in a particular way. Who's going to actually police it? I don't know, but at least we could do that and drive an understanding of those principles in general.
Well, ladies and gentlemen, unfortunately, our time is up. And, while we still had a raging and quite stimulating discussion. So I want to take the opportunity to thank you very much for this stimulating discussion. And I hope we can find ways to actually stay in touch and really find a way to, find solutions to work together to overcome some of the challenges that we've identified here together. So on that note, I wish you a wonderful afternoon and a good day for you, Delali, from Houston. Thank you.
Thank you, Soli and colleagues. Bye-bye.